In last class, we have studied the typical dicot embryo. Today, we shall continue the monocot embryo. Grass has taken as an example. Actually, or strictly speaking, grass seed is a type of fruit we call caryopsis. But generally, we talk like seed, grass seed. The structure of monocot embryo. To study the structure of monocot embryo, we have taken LS of grass seed or the fruit. In LS, the grass seed shows following details. The monocot embryo consists of only one cotyledon, or you can say the monocot seed consists of only one cotyledon. The dicots have two cotyledons, whereas the monocots have only one cotyledon. It is one of the morphological difference between monocot and dicots. So monocots have one cotyledon. Example for monocots: jawar, bajra, wheat, maize, etc. The grass or the our food crops belongs to the family Poaceae. Poaceae. It is grass family, formerly known as Graminae. Now it is changed to family Poaceae. If you take LS of grass seed, examine under the microscope, it shows only one cotyledon. In monocots, instead of cotyledon, we call scutellum. What is scutellum means? It is nothing but the cotyledon of monocots. If we examine the diagram, the scutellum is situated or found towards one side of embryonal axis. See the diagram. The scutellum is towards one side of the embryonal axis. Next, if we examine the embryonal axis, at the base, it shows what is known as radicle. Radicle. And it is protected by root cap, actually. Root radicle and root cap. This radicle in monocots is also protected by a covering or the sheath. We call it as coleorhiza. Coleorhiza is nothing but the protective sheath or the layer of radicle. The region above the level of scutellum is called epicotyl. Now you refer the diagram. The region found above the cotyledon or the scutellum is called epicartil, which shows shoot apex and leaf primordia. The young leaves are called leaf primordia. The leaf shoot apex is protected by another sheath just like root called coleoptile. It is a protective sheath of shoot apex. Thus, the monocot embryo shows shoot apex and root apex. Shoot apex is protected by coleoptile, whereas the root apex is protected by what is known as coleorhiza. Now you see the diagram and identify different parts. The diagram is important for CET examination. In NEET, mostly the diagrams are not given, but in CET, the diagram is very important. Okay, that's all about a monocot embryo. So we have seen dicot embryo and monocot embryo. Note the differences also. Differences between monocot and dicot. In monocots we have two cotyledons, whereas in dicot we have two cotyledons, and in monocot only one cotyledon. Then the embryonal axis, it is protected by a sheath. In case of monocot, we call it as coleoptile, and the base, the root tip, it is protected by another layer. We call it as coleorhiza. So we don't find this in case of dicots. Okay, scutellum is also absent in dicots. So that's all the difference between monocot and dicot embryo. Now we shall pass on to the next one. See the diagram. Next part is seed. In angiosperms, the seed is the final product of sexual reproduction. What is the meaning? After sexual reproduction, first the seed is formed. That's why it is called the final product of sexual reproduction. Then, what is seed? A very important question. The seed is defined as fertilized ovule. Fertilized ovule is called a seed. Okay. Seeds, if we examine, are always found inside the fruits. ಕಿತ್ಲೆ ಹಣ್ಣು, ಮೋಸಂಬಿ ಹಣ್ಣು, ನಿಂಬೆ ಹಣ್ಣು, ಮ್ಯಾಂಗೋ, ನಾವೆಲ್ಲ ನೋಡಿದಾಗ ಸೀಡ್ಸು ಹಣ್ಣಿನ ಒಳಗಿರುತ್ತೆ. It means the seeds are always found inside the fruits. Then, the structure of a seed. Seed. It's also very important. the seed structure. If we examine the typical seed, say you may take a dicot seed, you may take a bean seed. Very good example, bean. Typical seed consists of a protective layer. It means the seed is protected by a layer. What we call the seed coat. Next, the seed contains cotyledons. Two cotyledons are there because it is a dicot seed. And also at the center, there is embryonal axis. In a seed, we have protective layer seed coat, cotyledon, and embryonal axis. The seed coat, as I already told you, is a protective layer. It protects the seed from infection, from UV rays, etc. This seed coat is differentiated into two layers, outer testa and inner tegmen. What are these? Testa and tegmen. Testa is nothing but the outer layer of the integument. I told you already earlier. The integument forms a protective layer. The dicot ovule usually consists of two integuments or two layers, you can say. Outer integument or outer layer and inner layer. This outer layer transforms into what is known as testa. It is thick and protective layer. Protects the embryo. Whereas the inner layer we call tegmen, which is compared to testa quite thin. So thus the integument is transformed into testa and tegmen. The cotyledons, they store food material and they become thick. Okay, the function is storage of food material. Dicots have two cotyledons. You know that. Whereas the monocots have only one cotyledon. You can see the diagram of a dicot seed. Two cotyledons also. Mostly it is a bean seed because the shape of the seed is bean-like or kidney shape. Okay. Next. Classification of seeds. Seeds are classified into two different types. How they are classified? Based on the presence or absence of endosperm. What is endosperm? You already knew about this one. It is a nutritive tissue. So, based on the presence or absence of endosperm, seeds are classified into two types: albuminous seeds and non-albuminous seeds, or endalbuminous seeds and exalbuminous seeds. Okay, We shall see this one also. Albuminous seeds. These are type of seeds which contain endosperm. The albuminous seeds contain endosperm. See, the members of a family Poaceae. Say, for example, you take wheat, take maize, take barley. These are very good examples. Seeds which contain endosperm. Okay. If you examine the maize seed, I told you maize is not a seed, it is a fruit called caryopsis. What you call? Caryopsis. Ragi, navne, sajje, jola, godi, mekkejola. All of them are commonly called grains, but it is wrong. Biologically, they are fruits, what we call caryopsis. See the diagram of LS of maize seed, we come across the presence of endosperm and another layer, what we call aleurone layer, a protein layer is found in the maize seed. Dicots also have endosperm in the seeds. Very good example is castor. Castor means ಔಡ್ಲ. ಔಡ್ಲದಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮು ಇರುತ್ತೆ. Non-albuminous seeds, or we call exalbuminous seeds. The seeds without endosperm are called exalbuminous seeds or non-albuminous seeds. You know why the endosperm is absent in these seeds? Means the endosperm, the nutritive tissue, is completely utilized during the development of an embryo. That is why in the embryo we don't find the endosperm. But endosperm is present in albuminous seeds. Okay. Example for non-albuminous seeds: pea, groundnut, green gram, horse gram, cowpea. ಎಲ್ಲ examples. Note the differences also. Okay. So that's all about albuminous and endalbuminous seeds. Sorry, non-albuminous seeds. Now, one more important point is there from examination point of view is perisperm. What is perisperm? You know, when the ovule is young, it contains megasporangium or the nucellus. This nucellus is also a nutritive tissue. It also stores reserve food materials. So this nucellus, after the seed formation, in some plants, remains as a thin layer. It is called perisperm. What is perisperm? The persistent nucellus in a seed is called perisperm. Good example means gymnosperms are very good examples. The seeds contain perisperm. The angiosperms, some angiosperms, say for example, the black pepper seed, the beet seed contain perisperm. So that's all albuminous and non-albuminous seeds. Okay. Now we shall see or pass on to the next one. See the diagram showing two cotyledons and the protective layer. Now ಬೀನ್ಸನ್ನ ಓಪನ್ ಮಾಡಿದರೆ ಸಿಪ್ಪೆ ಬರುತ್ತೆ, ಹೊರಗಡೆ ಇರೋದು ಥಿಕ್ ಇರುತ್ತೆ, ಒಳಗಡೆ ಇರೋದು ಥಿನ್ ಇರುತ್ತೆ ಮತ್ತು ಶೈನಿಂಗ್ ಇರುತ್ತೆ, ಟೆಗ್ಮಾ. ಓಕೆ, ಸಿ ದ ಡಯಾಗ್ರಾಮ್ ಕ್ಯಾಸ್ಟರ್ which contains the endosperm. Identify this endosperm in this seed. Okay, and in castor seed a cap-like structure you find. See, it is nothing but caruncle, caruncle. Okay, at the same time see the LS of grass seed and different parts of the grass seed. See the LS of maize. See, endosperm is towards one side. The pericarp, thin layer is there. Then mesocarp is there. Endosperm is there. Endosperm is in large amount. See the diagram. And there is a layer. If you examine carefully, there we come across a layer of tissue. That is nothing but alluron layer, which is rich in a protein. That's all about the maize embryo. Now, the integuments of ovules are transformed into seed coat, already discussed. Micropyle of ovule remain as small pore in a seed. ನೀವು ಸೀಡನ್ನ ನೋಡಬೇಕು, ಬೀನ್ ಸೀಡನ್ನು ಸಣ್ಣ ತೂತಿನ ಥರ ಇರುತ್ತೆ, ಓಪನಿಂಗು, ಅದು ಮೈಕ್ರೋಪೈಲು. ವಾಟ್ ಇಸ್ ದ ಫಂಕ್ಷನ್ ಆಫ್ ದಿಸ್ ಮೈಕ್ರೋಪೈಲ್? ಥ್ರೂ ದಿಸ್ ಓಪನಿಂಗ್ ದಿ ಆಕ್ಸಿಜನ್ ಎಂಟರ್ಸ್ and which helps in the respiration of the seed. And also the water enters into the seed, which helps in seed germination. ನಾವು ಹೊಲದಲ್ಲಿ ಬಿತ್ತನೆ ಮಾಡಿದಾಗ ಬೀಜನ ಮಳೆ ಬಂದಾಗ ಬಿತ್ತನೆ ಮಾಡ್ತೀವಿ. ವಾಟ್ ಇಸ್ ದಿ ರೀಸನ್? ಫಾರ್ ಜರ್ಮಿನೇಷನ್ ವಾಟರ್ ಈಸ್ ಎಸೆನ್ಷಿಯಲ್. How this soil water enters into the seed means through this pore the water entered into the seed, then the seed swells. Because of the pressure, the seed coat ruptures and seed germination occurs. So the micropyle part is also a very important part. Okay. Then, ನಾವು ಹೊಲದಲ್ಲಿ ಹಾರ್ವೆಸ್ಟ್ ಮಾಡಿದಾಗ ಸೀಡ್ಸನ್ನು ಬಿಸಿಲಲ್ಲಿ ಒಣಗಿಸ್ತೀವಿ. ವಾಟ್ ಇಸ್ ದಿ ರೀಸನ್? ಸೂನ್ ಆಫ್ಟರ್ ದಿ ಹಾರ್ವೆಸ್ಟ್ ಅಂದರೆ ಹೊಲ ಕೊಯ್ದಾಗ, the seeds contain more moisture or more percentage of moisture. Some 20-25% of moisture is there. ಅಷ್ಟಿದ್ದನ್ನು ನಾವು ಸ್ಟೋರ್ ಮಾಡಿದರೆ, because of the moisture, fungus attacks. The whole seeds get infected. ಅದಕ್ಕೋಸ್ಕರ ನಮ್ಮ ರೈತರು ಭಾಳ ಬುದ್ಧಿವಂತರು, ಭಾಳ ಜಾಣರು, ಬಿಸಿಲಲ್ಲಿ ಒಣಗಿಸಿ the moisture is reduced 10-15%. Then the seeds are stored. Okay, ಯಾಕೆ ಸೀಡ್ಸನ್ನು ರೈತರು ಒಣಗಿಸ್ತಾರೆ ಅಂತ ಗೊತ್ತಾಯ್ತಾ ಈಗ? Then even if you want to export some seeds to some foreign countries, the percentage of moisture is very, very important. If the seed contains more than 15%, it is rejected. If it contains less than 10%, that seed also rejected. Very important. That moisture percentage must be some 10-15% only, within that. Okay, now, one question arises, whether the seeds are living or dead. ನೋಡೋದಕ್ಕೆ ರಾಗಿ, ಜೋಳ, ಸಜ್ಜೆ, ನವಣೆ, ಗೋಧಿ, ಮೆಕ್ಕೆಜೋಳ ಎಲ್ಲ ನೋಡಿದರೆ ಏನಪ್ಪ ಇವು ಬದುಕಿದವೋ ಸತ್ತಿದವೋ ಗೊತ್ತಾಗಲ್ಲ, ಹೇಳಕ್ಕೆ ಬರೋದಿಲ್ಲ ಅಂತ ಅಂದ್ಕೋಬೋದು. ಆಲ್ ದಿ ಸೀಡ್ಸ್ ಆರ್ ಲಿವಿಂಗ್. Very important points. The seeds contain embryo. Embryo is there in the seed. The metabolic activities also takes place because the seed contains living cells. But metabolic activities are at very slow rate. Okay, so that's all. Then one more point is there, dormancy. Dormancy. ನಾವು ತೆಂಗಿನಕಾಯಿನ ಮರದಿಂದ ತಗೊಂಡ್ರೆ ಭೂಮಿನಲ್ಲಿ ಹಾಕಿದರೆ 6 ತಿಂಗಳು ಆದಮೇಲೆ ಮೊಳಕೆ ಬರುತ್ತೆ. ಬೇಗ ಬರೋದೇ ಇಲ್ಲ ಮೊಳಕೆ. ಕೆಲವು 1 ವಾರಕ್ಕೆ ಮೊಳಕೆ ಬರ್ತವೆ. ವಾಟ್ ಇಸ್ ದಿ ರೀಸನ್ ಅಂದರೆ dormancy. The seed must pass the dormancy period. It varies from seeds to seeds also. Okay? ನೋಡಿ, ತೆಂಗಿನಕಾಯಿ ಇದು 6 ತಿಂಗಳಾಗದಲ್ಲೇ ಜರ್ಮಿನೇಟ್ ಆಗಲ್ಲ. ಅದರೊಳಗಡೆ ಬೇಗ ಮಾಡ್ಬೋದು by using scientific methods. With the treatment of some hormones, we can make the coconut to germinate early. Okay. So what is this dormancy? It is the inactive state of embryo, nothing but. The inactive state of embryo is called dormancy. Okay. That's all. So many points are there for NEET or CET examination.